Today we will continue talking about the control of eukaryotic genes. At the end of class, we were talking about transcription complex, which is involved, which you have the enhancers that have the activators that will attach, that will then combine with the transcription factors that bind to the promoter site and activate RNA polymerase to transcribe the DNA. So there's post-transcriptional control, which is alternative splicing, which will account for the variability of the protein families. So the exons will stay, and removing different introns will result in different mRNA pieces, which will then code for different proteins. So this alternative splicing accounts for the variability in the production of different proteins from the same DNA genetic sequence. You have the regulation of mRNA degradation. So the lifespan of mRNA determines the amount of protein synthesis. So the longer the lifespan of the mRNA, the more proteins that are synthesized. This can last from as short as a few hours to weeks, just depending on the mRNA sequence. This is something new that just came out. There's something called RNA interference. They're called your siRNA or small interfering RNAs. They're short segments of RNAs that are about 21 to 28 bases long. They bind to the mRNA and form a double strand and prevent the mRNA from being translated into proteins. It's almost like a death tag for the mRNA and triggers the degradation of the mRNA. So it'll break down. This causes gene silencing, so it prevents the gene from being transcribed, and this happens it's already been transcribed, but it prevents the gene from being translated into proteins. So you don't have the genes turned off, and you don't have proteins produced from this mRNA, because the small interfering RNA will bind with it, preventing the ribosome from reading the mRNA and creating the proteins. Well, the amino acids that create your proteins. So in this picture down here, so in the picture below, you have your siRNA or your small interfering RNA that will bind with the mRNA and create a double strand and prevent it from being translated in the ribosome. The action of the small interfering RNA. So you have your mRNA translation in the nucleus. Then you have a dicer enzyme, which will catalyze the production of the siRNA. So this will produce siRNA. The siRNA will bind to the mRNA and form a double strand. When it forms a double strand, there will be a breakdown enzyme called RISC, which is called RNA-induced silencing complex, and this will prevent the mRNA from being transcribed and will break down the mRNA. By breaking down the mRNA, you are turning off the gene. So RNA interference was discovered by Andrew Fire and Craig Mello. They found that these siRNA bind to the mRNA and prevent the production of proteins that create the different amino acids. They've used the siRNA as potential therapy for different diseases and cancers. So they want to try to use the siRNA to attach to tumor cells and silence those and prevent the creation and generation of tumors from the mutated cells. Control of translation. Blocking the initiation of translation, the regulatory proteins attached to the 5' end of the mRNA. It prevents the attachment of ribosomal subunits and initiator tRNA. This blocks translation of the mRNA protein. So if there is a blocking of the initiation of translation, the mRNA cannot bind and it can't be translated on the ribosome. So protein processing and degradation. Protein processing is a folding, cleaving, adding of sugar groups, targeting for transport. Protein degradation, there's ubiquitin tagging and proteasome degradation. So ubiquitin tagging, it's a regulatory protein found in most eukaryotic tissues. It signals the degradation of proteins through a proteasome. So what'll happen is the ubiquitin will join to the protein, and the proteasome will see the tag from the ubiquitin and will degrade the protein that is present, which you can see below in the picture. So ubiquitin, known as the death tag, marks unwanted proteins with a label. There's 76 amino acid polypeptide. It is 76 amino acid polypeptide. It labels protein. Labeled proteins are broken down into waste disposers by the protozoans. And these gentlemen below discovered the death tag back in the 1980s. So proteasome, it is a protein degrading machine. Cell waste disposer, it breaks down any proteins into 7 to 9 amino acid fragments. It is the cellular recycling center. So proteasomes, when ubiquitin tags certain proteins, the proteasome will come about and break down the longer polypeptide chains into a 7 or 8 amino acid fragment. So just a summary of gene regulation. In steps 1 and 2, you need to initiate and undergo transcription. During that time, you're looking at the DNA packing and the transcription factors. If the transcription factors are all present, it's going to undergo transcription. And with DNA packing, you want to ensure that the DNA has undergone acetylation. If there's a methylation, the gene is going to be turned off. Post-transcription, you're looking at the mRNA processing and the splicing and the alternative splicing that will result in different genes being turned on at different points. The poly A tail and the 5' cap will protect that mRNA sequence. If the siRNA is present, it potentially will bind to the mRNA and prevent it from being translated. During translation, you can block the start of translation. During translation, you can block the initiation stage, so there's a prevention of the ribosomal subunit and the initiator tRNA. So you're going to block translation of the mRNA. And then when you're looking after post-translation, there's protein processing and protein degradation. So if the proteins aren't processed correctly, or the proteins are broken down using ubiquitin as the tag, and the proteasome degrades the proteins aren't going to be present in order for the proteins to code for specific traits. Turn your question genes on and feel free to ask questions in class tomorrow. Have a good night. And for your academic pleasure, just for your own sake, it might be nice for you to be able to go through gene regulation and discuss or talk over each step for yourself and how it will affect gene regulation.